The Endosymbiont Hypothesis and Origins of Prokaryotes and Eukaryotes by Marcus Ford. Let us begin. Endosymbiont Hypothesis. The Endosymbiont Hypothesis, as you know today, was accredited to Lynn Margulis, who we'll talk about in a little bit. But let's start with just the basis. The endosymbiont theory states that some of the organelles that are found in the eukaryotic cells today were once prokaryotic microbes. According to the theory, the first few eukaryotic cells that were around displayed amoeba-like cell structure, in which they got their nutrients by phagocysts. These amoeba-like critters also contain a nucleus that was formed from a piece of cytoplasmic membrane cut off from the chromosomes. Through indigestion of prokaryotic cells and the eukaryotic cells survived within the organism and developed a symbiotic relationship. On another note, mitochondria formed when bacteria were capable of aerobic respiration were ingested. Chloroplasts were also formed when photosynthetic bacteria were ingested. Both of them eventually would lose their cell wall and much of their DNA due to them not having the benefit of being in a host cell where mitochondria and chloroplasts could grow. Alright, let's move on to the next slide. Who is Lynn Margulis? While the concept of eukaryotic cells being just a group of microorganisms was a common consensus first suggested in the 1920s by American biologist Ivan Wellen, it was Lynn Margulis who put all the pieces together and took it further through observations of mitochondria and chloroplast. The endosymbiotic theory of mitochondria and chloroplast was proposed by Lynn Margulis of the University of Massachusetts Amherst. In 1981, Margulis published a novel called Symbiosis in Cell Evolution, in which she describes that the eukaryotic cells originated as communities of interacting entities that joined together in a specific order. The prokaryotic elements could have entered a host cell through an ingested prey or parasite. Over time, the elements in the host could have developed mutually beneficial interaction, later involving to obligatory symbiosis. Margulis also claimed that symbiotic relationships are a major driving force behind evolution. According to Margulis and a fellow researcher named Sagan, life did not take over the globe by combat, but by networking. This observation led Margulis to consider Darwin's notion of evolution driven by competition to be incomplete. On to our next slide. Evidence in favor of endosymbiotic hypothesis. Mitochondria and chloroplasts are the same size as prokaryotic cells and both divide by binary fission. The mitochondria of some protists have Fs and homologs at their division plane. Mitochondria and chloroplasts also have their own DNA that is circular, not linear. Mitochondria and chloroplasts have their own ribosomes that are 30S and 50S subunits, but not 40S and 60S subunits. Several more primitive eukaryotic microbes, such as Giardia and Trichomonas, have a nuclear membrane, but no mitochondria. Alright, let's move on to prokaryotics and eukaryotics and their origin. Fossil records that indicate eukaryotics evolved from prokaryotes somewhere between 1.5 to 2 billion years ago. Two proposed pathways describe the invasion of prokaryotic cells by two smaller prokaryotic cells. They subsequently became successful included as part of now larger additional structures and capable of additional functions. We've already talked about endosymbiosis previously. Now let's move on to the next part of it, which is enfolding, membrane enfolding to be exact. The invasion of host prokaryote cell is theorized to be due to a host cell membrane being enfolded to surround both invading prokaryotic cells and thereby help transport them to the cell. The membrane in response does not dissolve but remain intact and creates a second membrane around the proto-mitochondrion and the protochloroplast. It is also known that in the modern eukaryotes, the inner membrane of both the mitochondria and chloroplast contains structures more similar to prokaryotes and eukaryotes, while other membranes retain eukaryote characteristics. And that's about it. I'm going to show you my sources before we go. Here's some of the sources I've used for my presentation. Thank you for sticking around, and I hope you enjoy the video. Goodbye.